is CORDIScovery. Hello, welcome to the special edition of CORDIScovery with me, Abigail Acton. This is the World Health Organization's Antimicrobial Resistance Awareness Week. So today we're talking to 3 EU projects that are looking at ways of tackling this vital challenge. Antimicrobials is the term given to antibiotics, antivirals, antifungals, and antiparasitics. The antimicrobial resistance, or AMR, organisms are developing, and our bid to counter that with new ways of controlling them is like an arms race, one that can be weighted in our favor if we use the antimicrobials that are still effective more carefully. The problem is complex, affecting human, animal, plant, and environmental health. Antimicrobials play a critical role in farming. Resistant microorganisms can develop in our food chain, moving between animals, humans, and the environment. Disturbing when you consider the World Health Organization estimates the global use of antimicrobials in the livestock sector alone will rise by 67% by 2030 to 105,596 tons. So what can be done to control the use of antimicrobials in livestock farming? And when it comes to plants, are there alternatives to the pesticides used today? While new solutions are being found, can we boost the way our current antibiotics work by making bacteria more vulnerable? Our 3 guests, whose work has been supported by the EU's Horizon 2020 programme, will explore the results of their projects to help answer these pressing questions. A warm hello to Anna Bejarano, who is the principal researcher on the RhizoTalk project. Anna is a postdoctoral researcher at the University of Trento. Her research focuses on understanding and exploiting soil microbial interactions and developing biopesticides and biofertilizers based on beneficial microorganisms for sustainable agriculture.
Hello everybody.
Hello, Anna. Fredrik Almqvist. Fredrik is Professor of Organic Chemistry at Umeå University in Sweden and co-founder of QureTech Bio AB. His research is aiming to develop new solutions for infectious and neurodegenerative diseases.
Hi, thanks. Really nice to be part of this pod.
Excellent. Nicolas Fortané, a sociologist at the French Research Institute for Agriculture and the Environment, based at Paris Dauphine University. Nicolas is interested in health issues that connect humans and animals, such as antibiotic resistance and infectious diseases. His focus is on socio-technical transitions towards sustainable agricultural systems. Welcome, Nicolas.
Hello, Abigail. I'm really happy to be here to talk today about this very important topic.
And we're very happy to have you. Anna, the RhizoTalk project looked into using microbial behavior in the soil to develop effective bacterial control agents. Can you break down this goal a little for us, please? What do you mean by bacterial biocontrol agents and why are these desirable?
Sure. The aim of this project is to look for alternatives to chemical pesticides to use more sustainable agriculture approaches to treat pests and diseases. It is well known for all of us that chemical pesticide is causing death to soil and leading to alarming outbreaks and also causing resistance to many pathogens are becoming resistant to such chemicals. Bacterial biocontrol agents are beneficial microorganisms that live in the soil and they interact with many other microorganisms and the plants. They represent a promising alternative to those chemicals because they produce many antimicrobial compounds and they can counteract those pathogenic microorganisms that are causing death to plants. They are not toxic, they are biodegradable, and they do not pose any risk to the human health or animals.
I see. So obviously that would be a better channel to explore. I think that I've heard the term bioactive metabolites. Can you explain what that means and why that's relevant? What does that entail?
Yeah, well, those bacterial biocontrol agents, they produce many bioactive metabolites that can be antibiotics themselves. It can be antifungal compounds, lytic enzymes that cause death to those pathogens, and also that they interrupt the infection of those pathogens. They can invade the pathogen and minimize their growth. They don't let them grow. And one of the problems with those bacterial biocontrol agents, while they are not widely used at the moment, let's say, it's because they work very well under control conditions. But when we go to the field, they need to interact with the indigenous microbiota and they need to adapt to new environmental conditions. They come, let's say, from very nice conditions in the laboratory where they are growing, the optimal conditions. But when they go to the field, we have heat, we have cold, we have wind, we have rain, we have many environmental conditions that might harm the activity of those bacterial agents, and also many other microorganisms that will fight or will encourage the adaptation of those microorganisms in the soil.
So they're challenged by the natural environment in ways that is difficult, I suppose. Well, are those ways difficult to assess prior to using them in the soil and in the real world? Can you identify what the challenges are likely to be for any particular strain of a biocontrol agent? So, for example, if you've got a promising biocontrol agent and you think you'll use it here, you know in advance the challenge for that will be X, Y, or Z?
Yes, actually, for example, by developing formulations, so let's say a mean of transport that protect them, that it can be a capsule or a liquid with many protectants, we can protect those bacteria against the environmental conditions. So also by providing particles that protect them against the UV light, the similar that we do when we go to the beach and we put some sun cream, we can do the same, treating these bacteria with kind of sun cream. So we can, by playing around with the ingredients of a formulation, we can protect our bacteria in a better way to minimize the dying from these environmental conditions. And then also knowing which are the partners that can enhance the adaptation in the soil. So which are my microorganisms that are present in the soil that will help me to colonize the plant and protect the plant against pests or diseases, we might encourage and we might enhance the use of a certain beneficial strain.
Right. And how I know also that in researching this area, one of the things that I found quite interesting is this notion of a quorum of bacteria. Can you explain what that means and tell us a little bit more about that notion?
Yes, exactly. This interaction is based on reaching a quorum. When bacteria are in a certain environment, at some point, if they want to coordinate a behavior, they start to grow to reach certain population. That's what we call a quorum. And when bacteria reach this density, they start to talk among each other. The same way that humans do, they have developed a language that is based on the production of chemical signals that can be detected by themselves and by other partners that are not from the same family. And they can this way communicate and coordinate, for example, the production of antimicrobial compounds. They can coordinate motility, so they will move towards certain
locations.
Location of the plant, and many other traits that are important for biocontrol activity.
And is this something that rather, as you were talking about, rendering the natural environment less problematic for the particular target microorganism, is this something that you can control in any way, or is there some way that you can release in certain environments? You say you identify existing microorganisms that could support the ones that you're targeting. Mm-hmm. Can you also work out if there's a quorum?
Yeah, we are working actually on that. The project was, my project was aimed to understand this, if we can use those signals to modulate the behavior when introducing a microorganism in the soil. And what we did was to expose a model bacterial agent to those molecules and know which were the molecules that are modulating or, let's say, promoting the biocontrol activity, and which ones were the ones that were blocking the biocontrol activity of our model strain. And then the next step was to select partners that can produce those beneficial signals in order to create a consortium and not only applying one bacteria at a time, but a group of bacteria, so that our model bacteria is already encouraged by neighboring or partners in a formulation. And actually working or having a focus on the quorum is something that is also very studied at the levels of human health, because bacteria can communicate, but they can also cancel the communication. So there are also some partners that will quench that it's how we call quorum quenching, will silence the communication among bacteria. This is also a way, for example, for pathogens that will impede the development of the pathogen.
And have you actually managed to work out how you could do this in the field itself?
Yeah.
What did you actually find?
Yeah, we are doing it in two ways. We are introducing these molecules in a formulation. So if we are developing, for example, a spray for spraying the plants, we are integrating this molecule within the spray that we will apply to the plants. Or in another way, we are introducing microorganisms that can produce these signals. So at the time we will apply our model bacterial strain and the other partner, once they get into the soil, the partner will start to produce this molecule, so it will activate the biocontrol potential.
This is absolutely brilliant. This sounds like such a great solution to current pesticides and quite heavy-handed ways of trying to control microorganisms currently. As you said, it's affecting soil health. Could you tell me a little bit more about how current controls are impacting on the environment?
Do you mean chemical?
Yes, I mean, when you say affecting soil health, in what way?
Yeah. At the moment, the major way to control pests is using chemical pesticides, although there are also other practices. Nowadays, there is a trend towards using integrated pest management practices that aim to use not only chemicals, but also crop rotation or understanding the weather conditions when there is going to be a pest outbreak in order to anticipate to the pest development. And the problem of using in a massive way chemicals is that they diminish the biodiversity of the soil. So a soil that is being treated with many chemicals has less population of beneficial bacteria. So in a first moment, let's say, it seems that it's beneficial because it counteracts the pathogen, it minimizes the pest and the disease development, but in a long term, it won't help because the beneficial microorganisms that also contribute to stop the pathogen development won't be there anymore.
This sounds very much like the human microbiome.
Yeah, exactly.
Yeah.
Actually, there are some soils that are very interesting from an ecological point of view and that we are trying to imitate, that they are called suppressive soils. Those soils are colonized by many beneficial microorganisms, and when certain pathogens arrive to the soil, they cannot develop. And it's very interesting, and we are trying to understand what's happening in this kind of soils to imitate what's happening in nature so that we can apply it in other soils that are not suppressive soils.
Are there a particular type of plant that grows in suppressive soils? I mean, could it be that the plants that are growing in the suppressive soils are producing some sort of chemical signal that prevents the...
It can be, not necessarily, but of course, also the plants produce molecules. They also produce some kind of communication molecules, of course, that can be not only detected by other neighboring plants, but also by all the microorganisms, the microbial community. Also, plants communicate with bacteria and bacteria with plants and so on. And of course, the kind of plant that is in the soil will determine the microbiome of the soil.
And are you finding one consistent species in these, what did you call them, the suppressed soils, the soils that are naturally free of these?
There are many soils that are called suppressive soils, and they are not all with a certain kind of plant. So
that would be too easy, wouldn't it?
Yeah, it would be.
Hey, everybody plant sunflowers everywhere and you won't have a problem anymore. Very interesting, Anna. Thank you so much. Thanks. Fredrik, your project has been looking at the impact of antibiotics and how they can be used to deal with bacteria by rendering the bacteria more vulnerable to them. And of course, you're also interested in the impact, therefore, on the microbiome of existing antibiotics. Would you have any questions for Anna, considering that overlap between the work that you're both doing?
Well, I think it has been fantastic to listen to, and and the discussion is very nice in that sense that there are many approaches today, of course, which relates to what Anna has also described. So quorum sensing is extremely important in pathogenesis when you have a pathogenic bacteria. So the question is, when is a bacteria pathogenic or not? So if you look at it, we talk about good and bad bacteria often in the public, but the bacteria turns bad if it ends up in the wrong place. So it can be, some bacteria can be of good in one place, but then pathogenic if it ends up in the urinary tract, for instance. So controlling when the bacteria is going to go into certain protective states, like biofilm formation is a typical situation.
What do we mean exactly by biofilm?
So biofilms is an extreme problem on the planet, meaning that bacteria, they clug together and form a very strong armory against the world, if we put it like that, and of course communicate among themselves. So they do this to protect themselves from the environment, and it can be the immune system if we're in a human, but it can also be conditions that Anna described. So biofilm, I mean, if you look at the hospital situation, it's typically if you get catheterized or, all foreign objects you get.
So hip replacements and things like that.
Right. You can imagine the modern healthcare. It's a lot of replacements going on, right?
Yeah.
And if you get those infections and if they are multi-resistant, then it's a really tough case.
Do you have any other questions particularly directed to Anna's research that she's been doing?
Well, it would be interesting to, as you touched upon, I mean, there are discoveries done, which is solely done because of the interplay between the bacteria in the microenvironment they are. I mean, and what you describe, Anna, is really nice in the way that you need a certain community to start to produce these active metabolites. So do you think, Anna, that we will find more new antibacterial agents by doing so? There was an article in Nature a few years ago where they introduced the so-called iShip technology. So they could, because some bacteria are not, you can't grow them without their friends, but they developed a technique to do so and also discovered new molecules that no one had seen before, natural products that are now promising new antibiotics. Do you see more of this coming from your work?
Yeah, I'm sure there will be many active compounds that will come and that we don't know yet. Actually, only in soil there are like 1% of the bacteria and fungi that are known. The rest are not discovered yet, or as you said, they are not cultivable. So this is something of a problem. We know who is there, but we cannot get it into the laboratory, cultivate it, and isolate these new antimicrobial compounds. There is a trend now to develop synthetic communities. So looking at metagenomics, knowing who is there with new molecular tools and using what we can say parts of soils directly, not culturing the soil, but introducing some soil to another soil in order to recreate it.
So when you say synthetic communities, what you mean by that then, I guess, is communities that are developed in a way that's highly manipulated.
Yes, that we know which are the representatives of a certain community in the soil. And with a collection of isolates, we try to imitate this community. So developing consortium based on, for example, 100, 300 different isolates. That, of course, it's a very simple way to imitate what is happening in nature, because nature is the quantity of isolates is much higher. But this way, once they arrive to the soil, they might enhance the population of the natural communities.
And isolates means, because you've used the term several times there.
I'm sorry. different bacterial strains or fungi.
Right. Okay, that's great. Thank you. So, Fredrik, to just ask you again a little bit more specifically about your project now, QTB4AMR is approaching the issue from a different angle. So you're looking at the efficacy of current generation antibiotics and how to make bacteria more vulnerable to them. First off, big question, of course, what does QTB4AMR stand for, please?
Right. So QTB is a shortening of QureTech Bio. So that is the company we founded a few years ago. A couple of professors coming together, realizing that we are doing research that can have an impact on and help people in the future. So we founded this company to be able to translate discoveries. And 4 stands for 4.
Of course.
And then AMR is antimicrobial resistance.
Right.
So,
okay. We've cleared that one up. Thank you very much.
Thanks.
So, I mean, a question that I would be thinking is, why is it not better to actually look for new antibiotics to which bacteria are not resistant? Why isn't pharma doing that? Or are they?
Well, So to be clear, it's definitely the approach that has been taken the most the past years is to try to find a new antibiotic with a new mode of action. That is, and it should continue. I mean, it's really important that we find new antibiotics with new mode of actions. But on top of that, it's also clear that we have antibiotics that have been developed and that we would like to use as long as they are efficient. And there are ways to make antibiotics more efficient again. There are so many things bacteria can do to be resistant. And if you then can come in with something that can make them vulnerable again, that would be fantastic. And there are, historical cases, but to my surprise, it's not developed that far and not really looked upon that thoroughly, I would say. So,
could you give me some examples of how bacteria can be rendered more vulnerable? I mean, I find the relationship between the resistant microbes and the antimicrobial battle to try and control them fascinating. How the relationship? It's a bit like an arms race. Tell me how you can undermine the bacteria to make them more vulnerable.
Exactly. So there are many mechanisms that antibiotics use and how they work. And a common target for many antibiotics is the cell wall, because that is the bacterial difference between eukaryotic human cells and bacteria. And but then there are, bacteria have different setup in terms of membranes. Some are called gram-positives and some are called gram-negatives. And the difference is how many membranes they have, if they have an outer membrane or not. And you have mycobacteria, which is the chief of all pathogens causing tuberculosis. A third of the world population is infected with tuberculosis, so it's an ongoing pandemic. And with this said, it's of course many things they can start to develop to be resistant. It can be a simple one, can be that they create or they have a pump that can efflux pump out the antibiotic. That is one thing. They can also develop, have enzymes that break down the compound, which is typically what happens for beta-lactams, the penicillins. You have so-called beta-lactamase enzymes that just chew up the penicillin. Uh-huh. So those are 2 examples, but there are many other things that you can do, and probably many things we don't know about that will affect the antibiotics mode of action. So
what do you do to the bacteria to cause them to not be able to function to protect themselves? Because that's what you're aiming to do, I think. Right,
and depending on the niche, we can also consider just disarming the bacteria, not killing it, and that would be even better from a pressure of developing resistance. I can take a short case. If you have urinary tract infection, for instance, the bacteria need to withstand shear forces because you pee, right? And mm-hmm. So they need to establish a colony in the urinary tract, and then they create the most amazing fibers called pili, and they are made like a helical structure, so they can be like a string and withstand the shear forces. It's absolutely fantastic. And they need these pili to hang on and start the infection. And but they are not needed elsewhere. So this is typically what we would call a virulence factor. They need it to cause disease. So if you then specifically can remove the function of this pili, you have a antivirulence compound.
Uh-huh.
And for some niches, antivirulence would be fantastic because especially for these diseases where you don't die the other day, and then we need antibiotics for those cases. I mean, when you have a severe sepsis coming in, you don't need you don't have time to even know what bacteria you're suffering from. So then the antibiotics are super essential.
Yeah. And I guess also what you were saying about the pili and the way that they provide an anchor or a way for these bacteria to hold on under the pressure of moving liquid. I guess those pili that you're targeting would not be necessary to bacteria that we would say friendly. As you said, it's not exactly the right term, but anyway, living in the microbiome, because obviously you want to be able to target elements of the hostile bacteria or the problematic bacteria while leaving the friendly, in inverted commas, bacteria intact. So that would be an example where you undermine the pili and that doesn't then affect the microbiome in the gut.
No, exactly.
Okay.
Another example we are working on is for tuberculosis. They transit into a very tolerant state. So when they are attacked by, you get them, it's airborne, so you get them into the lungs, and then the immune system attacks and form sort of inclusions. They are called granuloma. And in these granulomas, there is a war going on. So pH is lowered and it's an attack on the bacteria. But they survive this immune response and instead start to transit into a very tolerant state. So they change they don't divide that much, but they change their appearance, if I put it like that, roughly.
And how can we target that? How can we,
Yeah, so that Christina Stalling's group in St. Louis developed an assay to look at this transition, because also in this transition, they formed this biofilm we talked about earlier. And we screened compounds that could interfere with this biofilm formation. And then, so it was just looking at that phenomena rather than a specific target. And these compounds turned out to be amazing, and we have now been able to sensitize tuberculosis in the lab, not further than that, but still it was one of these moments when you, wow. So these bacteria, they get susceptible, even if they are resistant to the frontline antibiotic isoniazid. If you take a resistant bacteria that is resistant to isoniazid and combine it with this compound we develop, they are susceptible again. So it's really fantastic to look in the lab at least, and hopefully it can be translated to something.
Yeah. It must feel like a real breakthrough, and then it makes you wonder what other breakthroughs can also be achieved.
Right.
Yes, these kind of vulnerabilities must be fascinating to explore. I suppose it comes down to establishing exactly how the antibiotics work. PhysOrg reports on work being done by Professor Simon Foster from the University of Sheffield School of Biosciences. His team focused on MRSA and found that antibiotics caused small holes that span the cell walls, and that these holes got bigger as part of a growth-associated process, eventually killing the bacteria itself. They also identified some of the enzymes that are involved in making the holes. Is this the type of vulnerability you're interested in exploiting? For example, harnessing such enzymes to undermine bacteria?
I mean, exactly. This is one of these examples that new basic science is so important to discover. I mean, all vulnerabilities, if they can be boosted by something, I mean, is it a way to affect the regulation of these enzymes, for instance, or other ways? So to do that, you need to have a really strong basic knowledge. That's why basic science is so important.
And I believe...
And hope the public will appreciate that, because
the basic science is very important to understand how the world works around us, absolutely. And QTB for MAR, I believe that you were creating a method of bringing together chemical know-how to harness these vulnerabilities. Can you tell me a little bit more about the so-called platform that you've developed?
Right. It's fairly simple. It's when you want to change molecules and make look at different properties, you need to have the ability to fine tune and synthesize them in different ways, right? To change their properties. And to be honest, it takes quite a while to get those methods in place. So if you just find the molecule, like we do, many of the antibiotics have been discovered from nature, for instance, then it's very hard to start to change and fine-tune their properties. So we base our chemistry on more than 20 years of experience of making a certain type of molecules, and that is kind of a platform that we can quickly, when we do libraries and screen, we can quickly fine-tune their properties. Well, quickly is a
relative
term, but it's a relative term, exactly. So that is the way to explain it, that we have a well-established chemistry in the back, meaning that we can perhaps faster than otherwise push projects further and make them useful.
I see. So it's a little bit like a kind of an array of methods, and that you can lay your hand on the right method for the right context more quickly because of the way that these are organized. Is that a vague?
Yeah, it's a good way to see
like
it.
A library.
A molecule can be seen as you have kind of a center of the molecule, and then you decorate it in a simple way. And then what you decorate is actually creating the final product, right? So if you want to put something in the corner of the molecule and something in the right corner, and so it's really fascinating, to be honest. I mean, that's why I got hooked, and that's why
it does sound absolutely.
That's why I'm a nerd in organic synthesis.
Yeah, no, I could totally see why. Whether tweaking this bit over there will have this reaction, and if you do, will that happen? Yeah, no, absolutely. Does anyone have any questions here for Fredrik? Anna, maybe you do, given your overlap between the two concepts you're working on.
This is very interesting what you're working on. How do you explore this resistance? How do you really understand how you can make more resistant to antibiotics such an organism?
More vulnerable to antibiotics?
Yes.
The opposite.
Sorry.
Well, you have a point. You can also make them more resistant, so that is true.
Sorry.
No, but there are different ways to explore. First of all, what your compound does alone. So if you have a standalone situation, how will the bacteria respond? And if you know that the compound is creating a certain phenotype, meaning that the bacteria will have a certain setup, you can look for that and see if the bacteria get more used to the compound and can withstand the compound over time. So you take it in several rounds and see if resistance appears. But for these cases, when we have a non-antibiotic compound just disarming, we have done that a couple of times, like for bacteria that you may look at in the soil. Listeria is a typical problematic bacteria that lives in the soil but can be translated into the food chain, right? Mm-hmm. And we have compounds that can take away their ability to infect if they come into the humans or animals, but this compound have no effect on their living in the soil. Having fun in the soil is not the problem with these compounds.
It's fascinating, the ability to fine-tune so well and to target so well, affect it in this context but not in that context.
Yeah, that is the future. It's like the precise medicine. But I need to stress that it works in certain niches, and infection is very, very, if we take chlamydia, it's typically a situation where disarming would be fantastic, because you don't die from a chlamydia infection, at least not quickly, and it would be easy to diagnose. But today it's treated with normal antibiotics, and more than 140 million people on the planet yearly get treatment for chlamydia. So, and of course, that antibiotic should be used for these cases when you have sepsis and die within a day or two. So you have to find these niches when it still make a huge impact on the planet, if we can find solutions for those bacteria as well. And the focus is it easily gets only on these killer bugs and situations when we also need to do research on other bacteria because we need treatments.
Anna?
Good, maybe molecular tools also work for this bacteria that you talk about, Mycobacterium tuberculosum. Like nowadays, there is a great focus on CRISPR and such kind of tools. Have you ever thought of using those with such a complex microorganism?
Well, it's to be honest, not within my expertise since I'm a synthetic organic chemist, but I can definitely see your point that I think it will open up many doors, especially more related to your field. If you can introduce a bacteria that outcompete Mycobacterium or so, that would be. But I don't know. It's a bit far-fetched to me, and
not exactly your field. Uh-huh.
Although Emmanuelle did her discoveries here in Umeå, and I know her, so it's the CRISPR-Cas9
you know the researcher that's been looking at this.
Yeah, it was developed in Umeå, so we have celebrated a lot her Nobel Prize, I can tell you.
Oh, fantastic. Nicola, your project Roadmap wanted to foster transitions, or still does want to foster transitions towards a more prudent antimicrobial use in animal production. Can you tell us a little about the current situation? We frequently hear that there's an overuse of drugs such as antibiotics in farming, but what is the extent of the problem?
Well, as you know, antimicrobial resistance is quite a big issue, something that we call usually like a global health and a one health issue, because actually, of course, the overuse of drugs in farming, and especially antimicrobials, is one part of the problem. But there are many causes and many consequences to that. And of course, overuse is also happening in human medicine, for example. What we can say, I guess, about the use of antimicrobials in livestock farming is that it's also quite diverse. In certain regions of the world, in particular in Europe, reduction of antimicrobial use is on the way, but it's also a bit more complex than that, because we need to understand better what is reducing, where exactly it is reducing, and what would have a greater impact, because of course we can use antimicrobials for preventive purpose, for therapeutic purpose, in other regions of the world for growth promotion purposes, and all of that doesn't have the same impact necessarily on antimicrobial resistance. And also we need to be aware that worldwide it is expected that the use of antimicrobials in livestock farming is going to grow over the next year, over the next decade, because the demand, especially in meat, is rising.
Yes, I understand that the World Health Organization estimates that the use in livestock sector alone will rise by 67% by 2030.
Yeah, absolutely.
It's quite phenomenal, really. So why, Roadmap was setting out, I think, to change perceptions and to identify historic reasons and to use that information to change perceptions currently. So why is this such an area where there is so much dependence? It's a big question, but if you could sum up some reasons for how this has come about as a problem.
Yeah, there is, as you said, there is a very long story here, and what I'd like to stress first is that you used a very interesting word, like you talked about dependence, but we're talking here about a systemic dependence. This is our food systems and our health systems that are structured in a way that they rely so much, they have relied at least so much on the use of antimicrobials. And there is an English anthropologist called Claire Chandler who speaks about antibiotic as a quick fix, as a way to easily fix our need in productivity, profitability, and things like that regarding the way we produce food, the way we heal and cure people. And antibiotic, as she said, are infrastructure, our social infrastructure. And this is why, this dependence that you mentioned, I really consider it is a systemic one. And of course, there are perception, behavioral aspects of the overuse of antimicrobials. I'm not saying that it is not something that we can also target and that we could work on, but it could never work if we don't address all the structural factors in terms of what are our farming systems, how the veterinary drug market works, what is the structure of the food chain, how is the demand regarding meat and other animal products. And if we don't try to work on all of that, basically we won't reduce our systemic dependence on antimicrobials.
And what is Roadmap's role in trying to work on all of that? Because I know that the project is multi-pronged. You are trying to address different angles, different levels. Tell me a little bit about the work of your project.
Yeah, of course. Roadmap is trying to encompass these various aspects, these various issues. I mean, first of all, we have a series of tasks trying to understand the various factors that have at least, develop this massive use of antimicrobial and both structural and behavioral factors. So, as I said, structure of the food chain, but also the practice of the stakeholder, the cultural aspect, the knowledge. And when we are implementing quantitative and qualitative survey to better understand.
Who's participating in the surveys? Who are you asking?
Oh, we are 17 partners in about 10 countries in Europe, and we are undertaking surveys in 10 countries.
But is it farmers, or is it vets, or is it farmers and vets, or who?
So on this farm level aspect, yes, it's farmers and vets and some livestock technicians. But we also have surveys and trying to understand the strategies of some industrial stakeholders because what they do matters as well. So the pharmaceutical company, the agri-food companies, and we are also trying to assess the impact of some national action plans.
And what are you finding? What's coming through? I mean, maybe it's early days, but do you have some overall kind of image, ideas coming through?
Yeah, sure. I mean, the project is still, we are midway to the project. We don't have the final results.
Sure.
But,
any trends emerging though?
Yeah, first of all, I think this awareness that everyone is calling upon about the problem is here. Like, of course, this is sometimes it's a bit diverse, but vets, farmers, also the industry is aware of the antimicrobial resistance issue. And especially in Europe, we are already in a trend of reduction antimicrobial use. But of course, there are policy styles, policy cultures that diverse in the north of Europe, the south of Europe, the west of Europe. So we don't bring up the same solution, the same way of implementing the solution. And of course, the impact could be slightly different depending on the countries. What we also see is a development of quality labels by the food industry that is trying to reduce antimicrobial use, but that has also, which does actually reduce antimicrobial use, which contributes to this reduction, but which also has other effects or impacts on the structure of the food chain or on the food market in terms especially of concentration of the chain, industrialization or intensification of the practices. So it is still a challenge how the reduction of antimicrobial use will contribute or not to agroecological transition. And for me as a sociologist, I will always try to keep in mind and to communicate on the fact that, of course, reducing antimicrobial use is an objective that we must achieve, but maybe it is not the only or the final objective, because the real objective is this transition towards a more sustainable way of producing our food. And we need to find solutions to reduce antimicrobials that also have an impact on this transition towards sustainable agriculture.
It's interesting that you're talking about sustainable agriculture, because actually that brings us back to the work that Anna's been doing in the RhizoTalk project, because she's been looking at sustainable alternatives to the current use of pesticides by using microorganisms and the way they intercommunicate and interact as natural ways of reducing the number of problematic microorganisms in the soil. Anna, would you have any questions for Nicolas?
Yeah, actually, if I'm not mistaken, I think there are some trends now to use water derived from farming, like, for example, from pig farmings to be used in agriculture as a way to stimulate crops, so as biostimulants because of all the manure and so on. And I'm aware of, I heard or read that, actually the use of antimicrobial compounds is a bit of a problem if there are some residual amounts of those in these waste products, because then they will come to the crops and they will spread all around. And do you know if there are any kind of treatments in order to silence these antimicrobial compounds in the waste, or how can we get rid of them in order to use circular
Well, that's a very interesting question. I mean, unfortunately, you are probably not asking this question to the right person, because I'm not a biologist and I'm not perfectly aware of all the technical aspects of how basically we can remove these compounds from the waste and stuff like that. But it is definitely, I know that not in roadmap, but in other projects, like working on developing alternatives as well to antimicrobials, or trying to develop techniques or technologies that are trying to prevent the spread of resistant genes in the environment. Because especially AMR in the environment is basically, for me, I think it is a new topic, the new issue, that something that we have missed for decades, and that is relatively new. I mean, probably in the last 10 years, but we have focused so much on the spread and the dissemination of these genes between human and animals, and forgetting like the third pillar of the One Health concept, which is the environment and the ecosystems. And AMR is such a impressive, topic that we must address in such a way, and I think we are just starting to do it. So the question is very relevant to my opinion. Yeah.
Excellent. Well, thank you very much indeed. And thanks to all of you, actually, for this very interesting debate, very topical, given the fact that it's the World Health Organization's week of antimicrobial awareness raising. The work that you're all doing is obviously very important. And I wish you good luck going forward with all of it. Thank you very much for participating today and for giving us your time.
Thank you to you. It was very nice to participate in such a podcast.
Excellent.
Very interesting to meet you all.
Thank you, Anna.
It has been a pleasure. I mean, this discussion is super important, and it's really fantastic to see how different disciplines contribute to this very important task. And we haven't really touched upon it, but climate change that we all discuss is, of course, also impacting this situation. And Anna, you touched upon it, and it's for sure also interconnected with antimicrobial resistance. So,
as it's interconnected with absolutely everything.
A lot to look forward to.
Yes, indeed.
Yeah, thank you very much as well, and I'm very happy to have contributed to this podcast, and I think it's very important that we can talk about this such an important issue.
Well, maybe we'll have another one next time. The World Health Organization has another awareness raising week. Thank you, Nicolas, for joining us. Thank you, all of you.
Thank you very much.
Thank you very much.
Super. Bye-bye.
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 Are you interested in what other EU-funded projects are doing to tackle AMR? The CORDIS website will give you an insight into the results of projects funded by the Horizon 2020 programme that are working in this area. The website has articles and interviews that explore the results of research being conducted in a very broad range of domains. Whether you're interested in CORVIDs or COVID, there's something there for you. Maybe you're involved in a project or would like to apply for funding. Take a look at what others are doing in your domain. So come and check out the research that's revealing what makes our world tick. We're always happy to hear from you. Drop us a line, editorial@cordis.europa.eu. Until next time.